Hi all, welcome back to my channel. Today I am going to give you a very simple introduction on production of enzymes. Enzymes are macromolecular biological catalysts. They can accelerate or catalyze various chemical reactions to produce desired products from substrate. The first enzyme produced industrially was the fungal amylase, takadiastase. It was employed as a pharmaceutical agent for digestive disorders. Then types of enzymes. Microbial cells produce a variety of enzymes. Enzymes, they may be adaptive or constitutive enzymes. Constitutive enzymes are always produced regardless whether the particular substrate of enzyme is present in the medium. Adaptive enzymes are produced only in response to the particular enzyme substrate in the fermentation medium. Then based on the location we can see enzymes exocellular or endocellular. Endocellular enzymes are produced within the cell and are not liberated to the fermentation medium. Exocellular enzymes are liberated to the medium. Most enzymes produced by microbial fermentation for the commercial purpose are exocellular enzymes. Commercial enzyme production utilizes mainly fungi, bacteria and yeast, and the microbial strains we are selecting, they must be having a high yield capacity, that is strains with high enzyme yield capacity and genetic stability under the particular conditions of fermentation are normally selected. The methods employed. Two methods are employed for the commercial production of enzymes: semi-solid culture and submerged culture. First we can see semi-solid culture. Enzyme producing culture is grown on the surface of a suitable semi-solid substrate. The substrate consists of moistened wheat or rice bran. It must be supplemented with nutrient source. The production medium, it is prepared by mixing the bran with a solution containing any dissolved nutrient salt. The desired pH for optimum growth is adjusted with an acid. Normally the pH is kept at neutral level. Then the medium is sterilized in an autoclave. And the sterilized medium, it is spread on metal trays up to a depth of 1 to 10 centimeters under aseptic conditions. And the inoculation, it can be done either in the autoclave after cooling or in trays, and fungal spores are inoculated. Then a series of trays are inoculated and these trays are enclosed in a large vessel. Aeration, it is ensured by the circulation of humidified air over the surface of the culture. It is necessary to keep the temperature within the narrow limit. Heat generation occurs during the fermentation process, so the trays may be equipped with a cooling system. Extraction of enzyme is performed with water. These are some of the enzymes produced by semi-solid culture method. The precautions are: aseptic conditions must be maintained, escaping of spores to surrounding environment must be prevented. Advantages: it has low investment, high yield, it helps to cultivate the molds which cannot be grown in fermenters. Disadvantages are: more space is required, more labor, risk of infection is high, and the modern control methods cannot be applied. Next is submerged culture method. In submerged culture method, we are using a fermentation equipment. It is a cylindrical tank of stainless steel and the tank is equipped with an agitator, an aerating device and a cooling system and various other equipments, that is equipments for form control, monitoring of pH, temperature, oxygen tension, etc. will be present. And the good growth is not enough to obtain a higher enzyme yield. The production medium should be basically contain energy source, carbon nitrogen source, special growth requirements, etc. And in some cases, certain compounds present in the medium may induce or inhibit the production of the enzyme. Inducers are so expensive. For example, the presence of lactose induces the production of beta-galactosidase. And as the inducers are very expensive, constitutive mutants are usually used for the enzyme production. So, they do not require an inducer. Then glucose represses the formation of some enzymes such as alpha amylases. So the glucose concentration, it must be kept low. Certain surfactants in the production medium increases the yield of enzymes. Then most of the enzyme fermentation are carried out at neutral pH and the pH, it can be maintained at desired level by buffer addition, and the addition of non-ionic detergents such as 2-in-80 increases the yield of certain enzymes. Once the fermentation is finished, the fermented liquor, it is subjected to rapid cooling to about 5 degrees Celsius in order to reduce the deterioration of enzyme. Then after fermentation, the microbes are separated by filtration or centrifugation process. The colloidal particles can be separated by adding suitable coagulating agents. Suspended solid particles are removed by vacuum drum centrifugation or by filtration process. Extraction of endocellular enzymes can be done by disintegration of microbial cells using a homogenizer or a bead mill. Then, in case of large scale operations, salts are preferred to solvents because of explosion hazards. Then advantages are it requires less labor and space, low risk of infection, automation is easier, high yield can be obtained. The disadvantage is that initial investment is very high. This is the representation, diagrammatic representation or the flowchart for the submerged fermentation process. Here we can see the inoculum is added into the fermenter. After fermentation process, the mycelium is removed. Then the removal of bacteria, removal of insoluble impurities. After filtration process, we can see the enzyme, it is standardized and packaged and is ready for marketing. This is only a very simple introduction of enzyme production, that is commercial production of enzymes. Hope you will understand the session. Thank you. Have a nice day.